Good morning, good afternoon, good evening to everyone who's here. I'm so happy that we have people here from all over the world, especially to people who are staying up late. I really appreciate you being here. And welcome to the first student cafe of the year. So if this is your first student cafe, um, I'll give a little explanation for what they're about. The student cafes are a chance for students to learn and discuss specific areas of ethnobotany with experts whose careers focus on that area. These discussions are organized by the SEB Student Committee, and their goal is to broaden students' perspectives and understanding of ethnobotanical work, to better inform their own research and career trajectory, and to build a professional network within their areas of interest. Um, and since this event is also open to everyone, including non-members, I want to highly encourage those who are here who may not be already be members of the Society for Ethnobotany to become a member. Especially for students, it's very affordable. And oftentimes you can ask if you're in grad school, you can ask your PI about helping to cover the cost of a membership. But there's a lot of benefits. You get a free subscription to Economic Botany, which is the SEB's academic journal. You are also eligible to submit your own manuscripts to that journal. You can you have the opportunity to be invited and present at the annual meeting or the annual conference. You'll also be eligible for travel awards and discounted registration costs for those conferences, access to members only events. You're also eligible for grants and awards that the SEB offers, which it offers some really interesting and generous awards for its students and members. You also have access to our newsletter, which is Plants and People. And you also have the opportunity to network with other people in ethnobotany. And this is really the only society to do that. So highly encourage you to seek out a membership if you haven't already. And with that, we'll go ahead and get to the reason we're all here. It's my pleasure to introduce Dr. Josh Kellogg. He is a professor of metabolomics at Penn State University, which is where I'm also a grad student. And yeah, he's going to tell us about his work and a little bit about his career path. And then from there, we'll just open it up I'll be moderating. So it'll be a shorter presentation, and then the rest of the time will be for Q&A, especially from students. So I'll be moderating questions from people in the chat, and it'll be kind of like a informal interview almost from there. But Josh, thanks for being here, and I'll let you take it away.
Thanks, Savannah. Thank you for the invitation, and thanks y'all for tuning in, whether it's morning or night, wherever you happen to be. I'm really grateful for the opportunity to chat a little bit, and then yeah, ask any questions that you might have about, I don't know, I guess whatever whatever pops into your head. So yeah, I'll just talk for a little bit about kind of my trajectory, how I ended up here in Pennsylvania, and a little bit about some of the work that we do. We'll keep it short, and luckily I'm a fast talker, so I want to make sure we have lots of time for questions and comments and stuff from you guys. So yeah, so he, as Savannah said, I'm an assistant professor here at Penn State. We've been here 5 years. Time flies. Yeah, and so I have a really great, diverse, dynamic research team, which I'm very proud of. We'll see some of their pictures kind of floating through the talk. But I'm not from Pennsylvania. I'm actually from California originally. So my kind of path, how I got here, I grew up in California. I'm a chemist by training, right? So I was a chemistry major at UC Berkeley. Not involved in plants when I first became a chemist. I was trained as an organometallic synthesis chemist, like something completely different and clearly nothing like what I do now. But when I was at Berkeley, it's some of the courses that I have taken that have really shaped my whole career. And two of the classes, their pictures are up here, that I took. One was a chemical ecology class, right? So all about how plants, insects, microorganisms are competing and communicating through small molecules. That was taught by Isao Kubo. And then I took another class that was on medical ethnobotany, taught by Thomas Carlson. And so these two classes really resonated with me. They were electives, they were not required, but I, absolutely loved both of them. And that, I came back to these classes later when I was trying to figure out what I wanted to do with my life, because I graduated and I realized that, maybe not surprising, organometallic synthesis was not what I wanted to do for the rest of my days. Intellectually very stimulating and challenging, but my heart wasn't in it. And so not having another path, maybe I went and found a job. So I worked at a biotech firm, Biorad Labs, for about 3-ish years. And I was in their protein chromatography research division. We built new plastic resins that pharmaceutical companies use to separate insulin from bioprocessing methods and applications and things like that. Again, really fascinating stuff, but my heart really wasn't in it. And so I was thinking about grad school, going to get some more education, and my thoughts kept coming back to these two classes, chemical ecology, medical ethnobotany, natural products, plant chemistry. Like, those really fascinated me. And so I was looking around for programs that were taking students and did something in this sort of realm. And I actually ended up moving out to Illinois. My advisor, Dr. Marianne Lila, she had a project working with Native Alaskan communities around berries. She does a lot of berry work, blueberries, cranberries, things like that. But this project was centered all around wild native berries and how communities were harvesting them, their attitudes around climate change and the berries and impacting their resources. And then we had questions about chemistry and health benefits that the communities were curious about. So we started this project in Illinois. About a year into my graduate work, she got poached. She's got scooped up by NC State, and offered me the chance to go with her to finish my graduate career over in NC State. So I followed her into the Department of Food, Bioprocessing, and Nutrition Sciences, where we kept working with Alaska Native communities. But this project, actually, I pivoted away from berries and into seaweed, macroalgae, right? So again, looking at the chemistry, looking at consumption patterns, and looking at health benefits of traditionally consumed seaweed species. From there, after I, wore out my welcome, no, didn't wear out my welcome. After it was time for me to graduate, I actually found a postdoc position just down the road at University of North Carolina, Greensboro, in the lab of Dr. Nadja Chek. She's a really strong natural product chemist. And so we did a lot of really interesting work. This is where I started working in metabolomics and chemical profiling, a lot of mass spectrometry, excuse me. And I spent a number of years there, I think about 4 years in Nadja's lab doing, being a postdoc and doing a lot of really cool research, until this position opened up here at Penn State. And I was fortunate enough to get the job and make the jump up to Pennsylvania and drag the family up into the coldish, I guess. And yeah, so like I said, that was in 2019. And so, we've been here ever since. And so, the beauty maybe of being a PI in a lab and in a university is that, some of the questions, some of the projects that I've worked on in the past, I'm now able in a position that I can kind of bring them back in and start building them up again, which I really like. Because it probably comes as no surprise to any of you that, the natural world holds vast promise for treating all sorts of health conditions, chronic diseases, infectious diseases, everything, almost everything we can think of, right? And, if we look at, hard, drug development data, like this bores out that more than half of all of our pharmaceuticals, whether over the counter or prescription, in the last 4 decades come from natural resources, right? If we look at anti-cancer drugs, that goes up to 80%. Even antibacterial drugs, we're looking at over 60% coming from the natural world, right? So we will tend to work on plants in our lab. Most of our work is on plants. And what's really curious is that, there's 300, almost 400,000 plant species they estimate globally, right? 28,000 of those have been recorded as being used as medicine across 90 countries. This was a study done by the Kew Botanical Gardens in 2017, 18, something like that, a few years ago. But 10% of those have had their chemistry investigated, have had any sort of follow-up. And so there's a huge potential. And so, we've been kind of building our lab around medicinal plants, very broadly focused. And so there we go. So if I think of like the 4 main areas that I would describe our research as being, the first is, one of them is natural product discovery, right? We tend to focus on Appalachian kind of plants because we're in the northern reaches of the Appalachian mountain range. And we look for new interesting chemistry, because I'm still a chemist at heart, for both infectious and chronic diseases. But to do that, we employ a lot of analytical techniques. And so there we go. So we do a lot of computational work and statistical work, and a lot of analytical chemistry around chemical profiling, metabolomics, modeling of the data to try and tease out what this chemistry might be, what these effects might be having on the cell or organ or organism of interest. And so that's been really one niche, one area of our work going forward. And another part is we're also really interested in the concepts of synergy, right? We know from plants that none of these compounds work alone. They're always working in tandem with each other, either on the same sort of pathway or complementary pathways. But for large parts, drug discovery, development, just, areas of research tend to focus on one molecule, one target, right? And so we're looking to develop analytical techniques to do this better, but also, probe different plants and different resources to see how we can better understand their synergy and their interactions. And then, of course, this is a part that I was able to bring back after having been kind of latent through my postdoc years. We didn't do a lot of ethnobotany in Naj's lab. We were more focused on chemistry, which is fantastic. But I'm really excited that we're able to start building these relationships again with communities, with researchers in both domestically here in the United States as well as internationally. And so we have several partnerships that we've been growing and trying to cultivate over the last few years. Some of them are still in their infancy, because as people in the ethnobotany realm, we understand that it takes time. So a couple of quick projects. I don't want to take up too much more of our time. One project that we're looking at is tuberculosis, an infectious disease. So we're partnering with Worcester Polytechnic Institute up in Massachusetts. We started looking at Artemisia, right? Because tuberculosis, despite everyone's best efforts, is still a very nasty disease. About 10 million people get tuberculosis globally a year, and it kills about 1.5 million people a year, right? So it is an extremely deadly disease. Before COVID hit, it was one of the most deadly respiratory infections that you could get still. So, right. And so when we look at Artemisia as a plant, right? Nope, I thought it was structured, no structure. Artemisinin is the well-known anti-malarial compound from this plant. But we saw that artemisinin, great for malaria. It's okay at killing tuberculosis. But the plant itself is really interesting and really effective in the lab at killing tuberculosis. And there were reports coming out of Africa that there were some doctors using Artemisia tinctures and extracts in the clinic to treat tuberculosis patients. And so their question, and the question that prompted us, was what's in Artemisia that's not artemisinin that could be potentially active against tuberculosis. So we've been hunting this down for a few years now. We've found a couple structures that have moderate activity, but we think that the real benefit of Artemisia, again, is in the synergy of all of these phytochemicals working together. And so we're still looking for additional structures, new chemistry, and then trying to weave how they work along pathways and see what they do. And then just recently, we've branched into other plants for tuberculosis. The sumac genus has shown really interesting promise in treating tuberculosis. This was almost a random kind of plant we sent our collaborator to test, and she was very excited by it. So we'll see where that leads us. We do also, in the infectious disease world, we look for antibacterials that kill bacteria, but we also focus on ways to disrupt communication within and between the bacteria, right? And so this involves this quorum sensing signaling pathway and seeing if we can shut this down without killing the bacteria, right? So we didn't find, but the signaling pathway was discovered 10 years ago or so. And so we've been looking at, again, plants and mushrooms, we'll say plants, kind of broadly, but looking at plants that inhibit this feedback system and limit the communication of the bacteria to hopefully tamp down the seriousness of the infection and promote the immune system to clear the disease, which would be fantastic. We also work on some chronic diseases, right? So there's a receptor that's found in our gut, in our small intestine and large intestine, that was originally designed for detoxifying chemicals that we might ingest, whether those are actual toxins or pharmaceuticals or any sort of foreign substance. But we found, or others have found, that this receptor actually plays a large role in inflammation response, antimicrobial resistance in the gut, and just general gut health, right? And it's also implicated in several different chronic diseases, irritable bowel syndrome, cancers, autoimmune diseases, and even retinal degeneration in the eyes. And so our project, or my student's project, has been looking at both medicinal and culinary mushrooms to see if they have an effect on this receptor in the body, as well as starting to look at traditional Chinese medicine preparations that come from her background as well. And one of our other projects, it was even her picture, Savannah's project, is looking at the ghost pipe, right? So this is an Appalachian plant, really interesting in its ecology. It's a heterotroph, a microheterotroph. It's a parasitic plant, but it has a history of being used for a variety of chronic health conditions. Right? We see here, this is the cover of cover of poetry from Emily Dickinson from the 19th century with ghost pipe like emblazoned on the front cover, right? And so Savannah's been working, interviewing folks who use the plant currently and seeing what they're using it for, how are they collecting it, how often, things like that. But what we realize is, looking at the literature, like, people are still using it, and it's actually had this kind of upswing in popularity in the last few years, but nobody really knows, does it actually work? Is it potentially toxic? What's in it chemically? And so we've been working on looking at the pain aspects of this. And so, moving into cell work as well as some animal work to see, does it actually modulate pain? Time will tell. We're still working on that part. That's Savannah's job. And so all in all, we cast a pretty wide net looking for different plants. Like I said, mostly focused on the Appalachian region. And North American plants, right? So we have a growing repository of 120 different species so far, give or take, right? And as I said, most of them are focused in North America, but one of the projects that we've been embarking on in the last couple of years is in North Dakota, right? And so this leads into some of the ethnobotanical work that we've been engaged in that I started actually as a graduate student. And we've kind of, I've been able to circle back to, right? And so we have that already. We talked about that. Cool. Right. Because, ethnobotany is all about building these relationships. And so this work, as I said, started interacting with traditional communities and native organizations back as a graduate student. So I started as my advisor and a couple other collaborators began this Global Institute for Bioexploration that promotes, as it says here, ethical, natural, product-based exploration. And the goal was to kind of merge or bring together at the table traditional knowledge practitioners with more molecular biology kind of chemistry-based researchers and see how we could communicate. That was really the goal. And so part of this was developing a round of these bioexploration assays that could be run anywhere in the world with very little capital input. We didn't need intense instrumentation or machinery, even electricity. We didn't really need electricity either. But they gave us preliminary indicators on the potential health benefits of some of these plants or other natural resources. And so we did workshops where, again, we were trying to bring people to the table, both domestically. We did this a little in Alaska and North Dakota, as well as internationally. So I was involved in workshops in a couple different countries, and the Institute did more after I moved on. I haven't done any recently, though, that I know. Yeah. So I was really fortunate that I was able to kind of restart some of this work and build these partnerships up slowly but surely, which I'm really excited about. And we've been working with the United Tribes Technical College most recently in Bismarck, North Dakota. There we are. Then we're in Penn State. There we go. Right. And so at the core of this project, we were asking, the folks at the college and the community around, what health concerns do they have? What's driving their worries about their health? What do they want to know about their plants, if anything? And we don't need to get too deep into it, but they were really curious about what some of their traditional plants could offer in terms of health protection, especially in gastrointestinal health, and cancer was a big question as well. So we've started collecting some plants and sharing knowledge back and forth. And that project's ongoing, and we're excited to see where that goes. There we go. So, of course, none of this science never happens in a vacuum. I have an extremely wonderful, smart, dedicated team that is very dynamic and fantastic. None of this would happen without them. So I'm incredibly grateful for all of them and the work that they do. We also have great partnerships. I'm really pleased that our partnerships reach from botanical gardens to other universities, to tribal colleges, to indigenous organizations, because it really builds, I think, a very strong research foundation and lets us do some really interesting work and some cool science. And answer important questions. And so, of course, and then we have gracious funding from a number of organizations that, knock on wood, will continue into the future. And that's kind of what I got. So that's a brief snippet of some of the work that we've been up to these last few years, and we'll see where it all goes, I suppose.
Awesome. Thank you so much, Josh. So now I just want to open up the chat to questions. If anyone has, especially any students have any questions related to Josh's career or research or anything, I'll be moderating the chat, and we can just go from there.
You're stunned into silence.
I know.
Fantastic.
Maybe I'll have a question to kind of get us started.
Sure.
Um, um, the Could you reiterate the antibiotic? So what about the quorum sensing um is new in terms of antibacterial antibiotic research?
Great question. Yeah, and unfortunately, with the time I put that in, and then this morning I'm like, oh man, we could spend a whole hour kind of talking on that one subject. So the traditional way that we attack bacteria, from Alexander Fleming and penicillin, back in the 1930s, right, is we try to kill them. We want compounds that kill the bacteria and leave them dead, and that's what we've been after, up until now. The problem with that is that the bacteria divide very quickly, and they're very flexible evolutionarily, and so they can overcome and build in resistance into their genomes to combat these antibiotics that we're developing, right? And that's why we have this resistance, and it's increasing in the news, and these resistant strains are becoming a bigger health burden, both in the US and across the globe. And so the thought has been, well, okay, rather than trying to straight up kill the bacteria, what others have found that started this, is that the bacteria, when they start growing, they are not toxic, they're not pathogenic, they're just kind of growing, living their lives, doing their thing. But they reach a certain density, a certain cell density, that suddenly they realized, oh, now we can like form an army, right? And then we can now attack our host, right? So at this critical density, we see them upregulating invasion proteases, right? So they can break down tissue, they release toxins, they start to form biofilms that make themselves harder to eradicate by the immune system or by antibiotics. And so the thought was, okay, well, if we could silence this communication, if they didn't know that they were surrounded by their friends, right, other bacteria, then they wouldn't have this kind of metabolic shift, and they would stay this, kind of quiescent, planktonic kind of, oh, we're just floating around being bacteria. And that's what this quorum sensing system is this is the feedback mechanism by which, when the signaling molecule reaches that density, the bacteria recognize, like, oh, there's enough of us around that we can start, being more aggressive. And so the benefit of trying this approach is that we're not killing the bacteria, so they do not evolve resistance to these inhibitors as quickly as they do, more traditional kill the bacteria antibiotics. And this also, some studies have shown that this also allows the immune system a better chance at kind of mopping up the infection before it becomes a really dangerous infection. Yeah, so it's a really different way of thinking about it, which is pretty cool. So yeah, that's where we're headed with that. We do still look
Yeah.
for antibiotics that kill them too. There's still a need for, lethal antibiotics. But I think having kind of a combination, a synergistic approach might be useful.
Yeah.
So, yeah, I have a question. So Josh, have you used Artemisia in tuberculosis treatment, and how far the success has been achieved, and which species and up to which altitude you get this Artemisia plant?
Fantastic questions, right? So we haven't used it. We have not used it in a clinical trial, because we're still at the phase where we're trying to find the chemistry. There was a paper published, as I said, from some groups in Africa that were using Artemisia afra, not annua, but afra, as in the clinic as an anti-tuberculosis therapy. And if I didn't see who asked that question, but if you send me an email, I can send you the paper and we can chat more. So the afra that we have been using, we actually had a collaborator down in Florida who grows Artemisia. So elevation wise, it's zero. It's Florida, right? So it's pretty much flat. But in Africa, they get it from a range of altitudes. I don't have that off the top of my head. So we've been looking at both Artemisia annua, and we've been looking at afra. The other benefit of afra has been it doesn't produce artemisinin, so it doesn't even synthesize that anti-malarial compound. So we're able to look at all the other chemistry that's going on in the plant without the clouding effect of having artemisinin present.
Thank you. Thank you. Thank you. That's good. Wonderful.
We have a question in the chat from Kay Brinson, who is wondering, how do you narrow down which plants to test, and what methods do you find are the most efficient for this?
Good question. Yeah, because as much as I would love to grab every plant we could ever think of, that would stress everybody out too much. And we don't have that, we don't have the money to do all that work. So we do a couple different ways. One is we look at previously published literature, right? There were a lot of studies that were done in decades past, especially like older papers where they just kind of, they grabbed a lot of plants and they screened them and said, oh, this is, this works against a bacteria, and it's just like a check or a minus, like, does it work or does it not work? And we find that a lot of these plants were never followed up with. Like, there was this instance of, yeah, they work, and then that's all. And this paper was from like the 70s, right? So we're talking 50 years ago. No one's really looked at it. So that's one way that we kind of comb through literature to try to find potential plants. For the on the ethnobotany side, of course, we listen to our partners and we ask, they tell us what plants they are interested in researching and what parts of the plants and things like that. So we listen to them on that perspective. And then once we have, and sometimes we just we have a list of plants that we just kind of keep, that we're interested in, that we think about, that we're curious on, like the sumacs that we're now using in tuberculosis. Our lab manager, it was growing in fields next to her property, and she just brought some in just to see what happens, and it's kind of spawned all of this. So it's a combination of kind of random choice, but we try to be a little more rational about the choices. So yeah, we use a bunch of different things to try and hunt down the plants that we want. And we definitely find some that were in literature, and we follow up with them, and like, we can't replicate the results. Like, so we're like, who knows? And that's the way research goes, I guess.
Thanks, Josh.
Thanks.
The person who asked the question said that was really cool.
Oh,
So
thanks. Okay.
yeah. Our next question is from Jacob Woodward, who asked, what are some analytical approaches you have used to assess plant extracts? Is it generally a mass spectrometry technique? Are there some techniques that work better than others, or are there new or more promising techniques?
Oh, there's always new and more promising techniques. No, that's a great question. Thank you. Thank you, Jacob. So one of the natural product work is long and hard, and it's a long road. And so some of the trickiest part first is identifying what in this plant is actually the active constituent, right? Plants synthesize thousands of compounds, and so trying to tease out which one is the one that's actually doing the work that we see is the trickiest part. So we've been adapting to use more of an untargeted, what we call metabolomics, which is like a chemical profile of a plant. Hopefully everything, as much as we can measure, at least. Usually mass spec based because they are more sensitive, but you can do NMR based metabolomics as well. And then from there we do a lot of statistics, a lot of machine learning sort of approaches to try and tease out and correlate how the profiles are changing as the biological activity of the extract is changing as well. So yes, most of it in our lab anyway is mass spectrometry based. We do a lot of metabolomics. That's the way that we found tends to be it hopefully gets us to the active constituents faster. So we don't have to go rounds of fractionation and testing and fractionation and testing. We can kind of zero in more quickly on what is the active, and then target that for identification or isolation if need be.
Yeah, and if I can add to that, Josh, to Jacob's question, as a student in Josh's lab, something that was new to me, I think that something that additionally that this lab does is, so that the typical way of isolating an active chemical from a plant extract is it's called bioassay guided fractionation, where you take a plant extract, you put it through an antibacterial assay, you maybe do a preliminary fractionation, and you see which extract was the most active. Like, okay, the antibiotic compound must be in this fraction. Then you take that fraction and you fractionate it again. You divide it, and you just kind of keep doing that until you've isolated the one compound that's doing that. And that's been really powerful for discovering some compounds. But there's several problems or limitations with that. And the biggest one is that only really works if there's one compound doing all of the activity. It also really only works if that compound is in high abundance in the plant, because as you further and further fractionate and divide that plant extract, low abundance compounds will just inevitably be lost in the fractionation process. It's also really inefficient, and you usually need to start with like kilograms of plant material, which isn't always feasible in in the case of like rare plants or things like that. So something that's I thought was pretty cool when I joined is that, and something the Kellogg lab does, is they take the bioassay data and the mass spectrometry data, and they put it into like a machine learning model. So trying to skip those multiple rounds of fractionation by putting it into mathematical models that will correlate variations in different analytes with increases or decreases in bioactivity. So it's ideally a more efficient way of isolating or identifying bioactive compounds in a way that doesn't bias towards only high abundance compounds or plants that you can get a lot of material from. So, are there any, we just have one from, I hope I'm pronouncing this right, Ibithiri Briggs, has a question. So with a lot of medicinal plants in the tropics in less equipped laboratories, are there handy analytic procedures for the identifying of bioactive compounds in plants? For the example he gave is minion. I've never heard of that for sequencing. So I guess minion must be like a handy sequencing technique for like under-resourced labs.
That's a great question, and I will admit I also don't know what minion is. I don't do any genetics. I'm a chemist. Like, what can I say? I'm a chemist. But we run into this problem quite frequently with collaborators in labs across the globe, really, that the sort of analytical equipment that that is frequently used for this sort of work is expensive. Like there's no way to sugarcoat it. It's expensive equipment. It takes a lot to maintain it. And it's a luxury, a privilege that not every laboratory, not every university has. And we understand that. There are there's different collaborations that can be established where there are core facilities that will run some of these samples for you, at either low or no cost. And then once you have that data, then you can turn it loose. There's a lot of online open source software and databases where you can start to try to mine the chemical data for known bioactives, like known structures that might be in the plant already. Or at least you can figure out like what sort of chemical class family they're a part of. So the real bottleneck is getting that first set of analytical data. And so, yeah, I'm not sure if it's the best answer I have, but what we've found is trying to partner with other universities, other organizations where someone who has the equipment, who has the instrumentation can run them and at least get the researchers in the other countries the raw data that then they can run with and do the discovery part themselves if they wish, or continue to partner to further the discovery process.
I have a
Sure.
follow-up question to that then. So I think you're right that a lot of times under-resourced labs partner with bigger labs in terms of analytical chemistry techniques. There's because they're not at the point where we have, like nanopore sequencers. I know, they're less than the size of a microwave now, and you can just have them on the bench top. We're not really there yet with analytical chemistry. And so it's still like, multi-million dollar machines. But something that can be a problem with that, and Josh, this is my next question for you, is how do you handle things like data sovereignty, or data ownership when, an under-resourced collaborator or lab gives you plant material to analyze for them?
Right. So several of our collaborations, I'm thinking of, we're working with a team in Ethiopia right now. They had a series of plants that they were looking at for potential anti-cancer effects. And they ran into this exact bottleneck where they could do the cell assays, they could collect the plants and all that. They could make extracts, but they didn't have the analytical capabilities that we have here. So they were reaching out for partners. And so before we even did any work, we spoke with the grad students and the faculty members, and then the two universities, it's a lot of legal stuff that I don't fully understand, but we made sure that in the document of this memorandum of understanding and these transfer agreements where that they retain ownership of the data, right? So we are collaborators in this. We're happy to help, but it is still their project. It is still their data. And so they retain control over what, if it gets published, if it doesn't get published, where the project goes. So, we work really hard to make sure that their sovereignty and their privacy is protected. Sometimes we've been in projects where the communities don't want to disseminate where the plants were collected, and journals often want GPS coordinates and things like that. And we've had to tell them, no, we got them in this broad general area, and that's all we can share. So we work really hard to try and respect. It's essential that we respect their privacy, their sovereignty, and their wishes around their plants and their data.
Yeah. I just wanted to ask, Ibithuri, did we answer your question? I know I kind of had trouble
Yeah.
understanding what you were saying. Feel free to unmute yourself as well. All right. If we didn't answer your question well enough, put something in the chat and we'll try to address it. Riley Johnson asked, do you ever consume any of the botanicals that you research after finding that they may contain useful compounds for health? Or do you forage in general?
Yeah, so I do some foraging. I do collect plants and make teas and stuff, especially fall's coming, flu season's on us, the new elderberry syrup is going to be ready soon, things like that. In terms of botanicals in the lab, my collaborator, one of our collaborators on this Artemisia project, absolutely swears by Artemisia supplements, for lack of a better term. And she recommended it for all sorts of infections. She takes it almost daily, I think. But she's one that's like never had COVID, and she attributes that to, the properties of Artemisia. So I haven't taken it yet, but the evidence that we have is, so far it's preliminary but compelling. So maybe flu season is a good time to start, boning up on my Artemisia supplements.
Yeah, Pennsylvania in general is a great place for foraging. I'm not from Pennsylvania originally, and Pennsylvania is the nation's top producer of mushrooms, and mushrooms are super abundant in our forest here. So people not only forage for like herbals, there's elderberry, there's, blueberry, berries and edible herbs and stuff like
Yeah.
that. But there's also a ton of mushroom foraging, and the mushroom foragers in Pennsylvania go hard. I was really surprised when I moved here, like how intense people here take their mushroom foraging. So it's definitely a thing around here. Um, yeah.
Yeah, yeah, yeah,
Um,
they are hardcore about foraging for mushrooms, and so it's very protective of their locations. They can be well guarded secrets.
yeah.
Yeah.
Um, are there any more? I don't have any new questions in the chat. Is there any more questions for Dr. Kellogg? I think Josh, I put your email in the chat because someone asked for it.
That's fine. Yeah,
So
if anyone thinks about questions or has things they want to discuss later, I'm always that way in seminars. I people are like, do you have any questions? I sit there like, no, I don't have any questions. And then like 10 minutes later, I'm walking back to the office. I'm like, oh, I should have asked this. So if something pops in your brain, yeah, feel free to shoot me an email and we can chat further about anything. Really.
yeah, I think everyone should in the chat. There is Dr. Kellogg's email in case you have any questions for him later.
I was
Yeah,
going to
are
ask a
there
question. You have an amazing background academically, and since many of the people attending might be students, and I've done the same where I started out in one field and then totally went in a different direction. It's kind of overwhelming in a way, and I'm wondering if people just want to open up and chat if they have questions about how to find the right professor, or how do you figure out kind of the topic you want, or how to combine things. It's a dance that we do with our lives and finding a job or finding a place we want to live, or an academic that we want to work with. And I didn't know if you had any suggestions along those lines.
That's a great question. Yeah, because as you saw in my, like little map timeline, I switched directions of careers a couple different times. And it's not easy. And, this is not to make myself sound too old, but the internet was not what it quite was today. So there's a ton of more resources out there, which can be both maybe a blessing and a curse, right? In terms of finding a mentor, um, especially in natural products and in ethnobotany, like we're a small group. Like there's not a ton of us in the even globally, but especially if we're thinking about in the United States, like there's not a lot of us here. So when I was looking, I was searching. I did some searching through relevant journals, looking for, people whose papers I thought was interesting, right? So looking at economic botany, looking at the Journal of Ethnopharmacology, Journal of Natural Products, like, that is one way to try and see what people are doing and what might be interesting to you. And then from there, it's, from going into graduate school, sometimes it's I always recommend just reach out and email, that's important. And then when you're choosing a lab, always talk to the students. I encourage that. I did that when I was looking for a graduate school. I chatted with the students in each of the labs I was applying to get a sense of, what is the PI like? There are some PIs that are very strict. They want you there at 8 o'clock and you work until you're done, and you better be here on a Saturday or else. There are other labs that have very different cultures and atmospheres. And so finding that is almost as important as finding a project that you like, is a PI and a team that you feel like is going to support you, is going to work with you and help you grow as a student, as a researcher, as a scientist. And that's ditto for if you're in graduate school, like maybe some of you are, and you're thinking like postdocs, if you're headed into academia, if you want to go down that road. Again, finding a good mentor is really key. And I'm lucky that, both Marianne, both Dr. Lila and Naj, Dr. Check, were fantastic mentors that really supported me. And I would not be here. I can say that truthfully. I would not be where I am now if it weren't for the two of them. But it's tough.
That's
And
awesome.
so even if you reach out to folks who are in your field, even if you don't want to work with them or they don't have opportunities or openings, like this sort of networking is going to pay dividends going forward, right? Because you could reach out to other grad students. Maybe they know a lab, maybe they know a PI, or they have a position or know something. You can reach out to more senior folks and ask questions and kind of get direction. So, this is something I wish I had been doing more of. When I was coming back from industry, so I was out of academia and university kind of life for a number of years. But this is what I would recommend.
That's really great. I think that's all good advice, Josh. That's really similar advice, as going into the 4th year of my PhD, that's very similar advice that I would give to anyone searching for the right grad school or even post-grad environment. So Jacob Woodward asked another question, and I think this is another really good one. How do you approach the ethical considerations of plant gathering and then constructing potential medicines from that? And could they be poached or over harvested if brought to the market? If they could be poached or over harvested, should we try to mitigate that?
Yes, definitely. So that's a huge question that we approach when we're thinking about, especially if there's a plant that is very promising that is going to move into animal studies, tox studies. We don't have anything in our lab that's moving to clinical trial yet, but going that route, the growth habits, the abundance of the plant is extremely important, because there's a number of plants that are being poached. Every year in North Carolina, they have folks that get nabbed in the Smoky Mountain National Park and along the Blue Ridge for poaching American ginseng out of the woods. They do not take kindly to that, especially on federal property. So those are definitely things that we have to consider before, we try to consider before it gets to the point where people would actually be out harvesting it. Artemisia is something right now that is not endangered. It's not really, knock on wood, at risk of over harvesting. And it's pretty amenable for growing in greenhouses and in controlled growth chambers and things like that. So for this particular plant, we don't have a worry. But Savannah's work on the ghost pipe, it is a much slower growing. It's not rare, but it's more uncommon. And there's definitely a risk of over harvesting. If we were to come out and say, yes, it cures your pain and it's fantastic, then that's something that we talk about, and we would have to think about real long and hard before widely disseminating maybe those kind of claims. Because we have to think about it from a conservation perspective as well as a human health perspective. There are labs that do that kind of work in terms of translating the biosynthesis from the plant into something like a yeast or a bacteria that they can grow in the lab very rapidly without any impact on conservation or environmental harvesting. So, but that can be a long road. So there's a lot that goes into that, but it's really important to think about that, especially if a plant is rare or has the risk of being endangered or over harvested like that. Yeah.
And I think a lot of it also comes down to your collaborator. If you're sourcing the plants from someone else, like the tribal college in North Dakota, like they're driving the bus as far as like what they want to research, what they want to tell us, like
how much
that can kind of
yeah.
Right.
Yeah. So we definitely, in those cases, we definitely trust our partners and respect, like Savannah said, respect their wishes on, how much we disseminate, how much can be harvested, where it is, things like that.
Yeah. We have a question from Nishant who says, do you have any tips for writing a good proposal to funding agencies?
Oh man. That that's a tough one,
Big,
right? You know, so
take a
it
deep breath there.
yeah, that was a deep breath. You know, I speak mostly from a US perspective, right? So we apply for funding from the National Institute of Health, the US Department of Agriculture, some from the National Science Foundation. And in the US, I will say very candidly that it's hard for natural products and botanical medicines to find good homes for funding. A lot of the times, this is not to be doom and gloom, right? We do have funded projects which we're really excited about, but a lot of times, you know, they don't want to fund screening projects. They say you're fishing, we're not going to support that. They really want you to know the chemistry or have an idea of the chemistry before they'll fund it, which is very counterintuitive because we need to do the screening and all this preliminary work to know the chemistry. But anyway, I think the key that we have found for funding proposals, one is to have a good hypothesis that's, you know, kind of universal for funding. Have a really good collaborative team in place. You know, so more and more agencies, especially with larger grants, they're not looking to fund one lab alone. You know, so we have a project that we put in for on machine learning and kind of AI sort of work, where we collaborate with another lab at our medical school. You know, we have other projects that collaborate with labs in chemistry or pharmacology or plant biology. You know, so reaching across to other disciplines and other departments like really helps strengthen those proposals and make sure you have the right expertise and you're not trying to just go it alone. And then make sure, you know, the other thing, as hard as it is make it relevant to the agency. You know, in the US, you know, the NIH puts out priority lists every few years. They have, you know, strategic plans and this, that, you know, things they want to focus on. And so being able to write your proposals so that you can say, yes, it addresses or partially addresses these topics that you said you're interested in, you know, definitely helps kind of whet their appetite and gets them excited.
Yeah,
And
I
then
would
be
say,
persistent. It takes a while.
yeah,
You know, we've had ones that we put in 3, 4, 5 times and still have not been funded. And that's hard when you put so much effort into that. So there is a bit of patience and a bit of yeah, patience that's involved.
yeah. I would say as a student, what I found to be really helpful is finding someone who has successfully applied for the grant you're looking at. Like it, so there's a lot of like government pre-doctoral fellowships for students here in the US, and like our college actually does whole like workshops on how to successfully apply for these big grants. But even with any fellowship, if you can't find a workshop or anything, um, reach out to someone who has successfully gotten this grant. Um, Even if they don't necessarily even go to your school. Like there's this grant I'm applying to now. I was actually like nominated to apply for it, but I don't really know anything about it. And so I like Googled the past awardees and found her on LinkedIn and like messaged her and was like, hey, uh, could you tell me more about this award? And I actually still haven't submitted it yet, so I'll let you know if that was actually a successful technique. But even like for peace of mind, like because like Josh said, these agencies are so specific in what they want. Um, and you kind of have to play the game a little bit of making sure you say the right things, or you know, you're kind of hitting um, you know, the topics that they're interested in. And so getting like an inside perspective on the agency or the specific award and what they want. I found that to be really helpful, at least in just helping me to write the application, to know, feel like I know what I'm doing.
That is a great thing. And if I can hop in, that reminded me. In addition to finding post-award winners, which is a great method, and even at the federal level, you can search. They have databases because it's public information of everyone they've funded for grad student fellowships or certain awards. So you can search by keyword and years and all this. You can also reach out to the agency themselves. You know, in the US, the NIH has program officers who are kind of liaisons who help people through this path. So you can send them your ideas and say, I want to talk about, you know, the toxicity potential of this plant. This is why I think it's important. And they're usually pretty good at writing back and saying, okay, you know, I think, you know, they won't tell you what to write, but they'll say, yes, this is something we would be interested in. You know, make sure that you're emphasizing, you know, think about X, Y, and Z when you're writing. Or they'll be honest and say, you know, that doesn't really fall within the scope of what we're looking to fund right now. You know, maybe try somewhere else. You know, so reaching out directly to some of the funding agencies, and you can find their contact info on their websites, is another avenue to help craft that proposal and make sure it's, you know, checking the right boxes and catching their interest. It's a learning process. I'll say that. I feel like I'm still learning. And we've submitted, you know, probably a dozen grants this year so far. So
Yeah, so we are, you know, we're past 11 a.m. We're over an hour now. Is are there any last minute questions for Dr. Kellogg? Otherwise, I'm thinking this might be a good place to stop. Going once, going twice. So, well, awesome. Thanks again, Josh, for taking the time.
oh, thank you everybody for great questions. You know, good questions always make me think too. So you've definitely given me more to think about what we do and how we do what we do. So thank you guys for comments and questions and having me on this morning or afternoon or evening or, you know, today.
Yeah. Yeah, well, I think we'll end it there. Thanks everyone for coming again, and making time in our diverse schedules to be here. Yeah, and I hope to see you. Our next cafe will be in October. The date is TBD as we kind of decide on the faculty member and work with their schedule, but it'll probably be somewhere at the end of October. So hope to see lots of you there for that one as well. Yeah, thanks everyone, and have a great rest of your day or evening.
Thanks everybody.